Section 16.6, Nervous System. By the end of this section, you will be able to describe the form and function of a neuron, describe the basic parts and functions of the central nervous system, and describe the basic parts and functions of the peripheral nervous system. As you read this, your nervous system is performing several functions simultaneously. The visual system is processing what is seen on the page. The motor system controls your eye movements and the turn of the pages or click of the mouse. The prefrontal cortex maintains attention. Even fundamental functions like breathing and regulation of body temperature are controlled by the nervous system. The nervous system is one of two systems that exert control over all the organ systems of the body. The other is the endocrine system. The nervous system's control is much more specific and rapid than the hormonal system. It communicates signals through cells and the tiny gaps between them rather than through the circulatory system, as in the endocrine system. It uses a combination of chemical and electrochemical signals rather than purely chemical signals used by the endocrine system to cover long distances quickly. The nervous system acquires information from sensory organs, processes it, and then may initiate a response either through motor function, leading to movement, or in a change in the organism's physiological state. Nervous systems throughout the animal kingdom vary in structure and complexity. Some organisms, like sea sponges, lack a true nervous system. Others, like jellyfish, lack a true brain and instead have a system of separate but connected nerve cells, neurons, called a nerve net. Flatworms have both a central nervous system, CNS, made up of a ganglion, clusters of connected neurons, and two nerve cords, and a peripheral nervous system, PNS, containing a system of nerves that extend throughout the body. The insect nervous system is more complex, but also fairly decentralized. It contains a brain, ventral nerve cord, and ganglia. These ganglia can control movements and behaviors without input from the brain. Compared to invertebrates, vertebrate nervous systems are more complex, centralized, and specialized. While there is a great diversity among different vertebrate nervous systems, they all share a basic structure: a CNS that contains a brain and a spinal cord, and a PNS made up of peripheral sensory and motor nerves. One interesting difference between the nervous systems of invertebrates and vertebrates is that the nerve cords of many invertebrates are located ventrally, toward the stomach, whereas the vertebrate spinal cords are located dorsally, toward the back. There is a debate among evolutionary biologists as to whether these different nervous system plans evolved separately, or whether the invertebrate body plan arrangement somehow flipped during the evolution of vertebrates. The nervous system is made up of neurons, specialized cells that can receive and transmit chemical or electrical signals, and glia, cells that provide support functions for the neurons. There is great diversity in the types of neurons and glia that are present in different parts of the nervous system. Neurons and glial cells. The nervous system of the common laboratory fly, Drosophila melanogaster, contains around 100,000 neurons, the same number as a lobster. This number compares to 75 million in the mouse and 300 million in the octopus. A human brain contains around 86 billion neurons. Despite these very different numbers, the nervous systems of these animals control many of the same behaviors, from basic reflexes to more complicated behaviors like finding food and courting mates. The ability of neurons to communicate with each other as well as with other types of cells underlies all of these behaviors. Most neurons share the same cellular components, but neurons are also highly specialized. Different types of neurons have different sizes and shapes that relate to their functional roles. Like other cells, each neuron has a body cell, or soma, that contains a nucleus, smooth and rough endoplasmic reticulum, Golgi apparatus, mitochondria, and other cellular components. Neurons also contain unique structures for receiving and sending the electrical signals that make communication between neurons possible. Dendrites are tree-like structures that extend away from the cell body to receive messages from other neurons at specialized junctions called synapses. Although some neurons do not have any dendrites, Most have one or many dendrites. The bilayer lipid membrane that surrounds a neuron is impermeable to ions. To enter or exit the neuron, ions must pass through ion channels that span the membrane. Some ion channels need to be activated to open and allow ions to pass into or out of the cell. These ion channels are sensitive to the environment and can change their shape accordingly. Ion channels that change their structure in response to voltage changes are called voltage-gated ion channels. The difference in total charge between the inside and outside of the cell is called the membrane potential. A neuron at rest is negatively charged. The inside of a cell is approximately 70 millivolts more negative than the outside, represented as minus 70 MV. This voltage is called the resting membrane potential. It is caused by differences in the concentrations of ions inside and outside the cell and the selective permeability created by ion channels. Sodium-potassium pumps in the membrane produce the different ion concentrations inside and outside of the cell by bringing in 2 potassium ions and removing 3 sodium ions. The actions of this pump are costly. 1 molecule of ATP is used up for each turn. Up to 50% of a neuron's ATP is used in maintaining its membrane resting potential. Potassium ions, which are higher inside the cell, move fairly freely out of the neuron through potassium channels. This loss of positive charge produces a net negative charge inside the cell. Sodium ions, which are low inside, have a driving force to enter, but move less freely. Their channels are voltage dependent and will open when a slight change in the membrane potential triggers them. A neuron can receive input from other neurons, and if this input is strong enough, send the signal to downstream neurons. Transmission of a signal between neurons is generally carried by a chemical called a neurotransmitter, which diffuses from the axon of one neuron to the dendrite of a second neuron. When neurotransmitter molecules bind to receptors located on a neuron's dendrites, the neurotransmitter opens ion channels in the dendrite's plasma membrane. This opening allows sodium ions to enter the neuron and results in depolarization of the membrane, a decrease in the voltage across the neuron membrane. Once a signal is received by the dendrite, it then travels passively to the cell body. A large enough signal from neurotransmitters will reach the axon. If it is strong enough, that is, if the threshold of excitation, a depolarization to around negative 60 millivolts, is reached, then depolarization creates a positive feedback loop. As more sodium ions enter the cell, the axon becomes further depolarized, opening even more sodium channels at further distances from the cell body. This will cause voltage-dependent sodium channels further down the axon to open and more positive ions to enter the cell. In the axon, this signal will become a self-propagating brief reversal of the resting membrane potential, called an action potential. An action potential is an all-or-nothing event. It either happens or it does not. The threshold of excitation must be reached for the neuron to fire an action potential. As sodium ions rush into the cell, depolarization actually reverses the charge across the membrane from negative 70 millivolts to positive 30 millivolts. This change in the membrane potential causes voltage-gated potassium channels to open, and potassium ions begin to leave the cell, repolarizing it. At the same time, sodium ion channels inactivate, so no more sodium ion enters the cell. Potassium ions continue to leave the cell, and the membrane potential returns to the resting potential. At the resting potential, the potassium ion channels close and sodium ion channels reset. The depolarization of the membrane proceeds in a wave down the length of the axon. It travels in only one direction because the sodium channels have been inactivated and unavailable until the membrane potential is near the resting potential again. At this point, they are reset to closed and can be opened again. An axon is a tube-like structure that propagates the signal from the cell body to specialized endings called axon terminals. These terminals, in turn, then synapse with other neurons, muscle, or target organs. When the action potential reaches the axon terminal, this causes the release of a neurotransmitter onto the dendrite of another neuron. Neurotransmitters released at axon terminals allow signals to be communicated to these other cells, and the process begins again. Neurons usually have one or two axons, but some neurons do not contain any axons. Some axons are covered with a special structure called a myelin sheath, which acts as an insulator to keep the electrical signals from dissipating as it travels down the axon. This insulation is important, as the axon from a human motor neuron can be as long as a meter, 3.2 feet, from the base of the spine to the toes. The myelin sheath is produced by glial cells. Along the axon, there are periodic gaps in the myelin sheath. These gaps are called nodes of Ranvier and are sites where the signal is recharged as it travels along the axon. It is important to note that a single neuron does not act alone. Neural communication depends on the connections that neurons make with one another, as well as with other cells like muscle cells. Dendrites from a single neuron may receive synaptic contact from many other neurons. For example, dendrites from a Purkinje cell in the cerebellum are thought to receive contact from as many as 200,000 other neurons. Figure 16.19. Neurons contain organelles common to other cells, such as a nucleus and mitochondria. They also have more specialized structures, including dendrites and axons. Everyday connection, neurogenesis. At one time, scientists believed that people were born with all the neurons they would ever have. Research performed during the last few decades indicates that neurogenesis, the birth of new neurons, continues into adulthood. Neurogenesis was first discovered in songbirds that produce new neurons while learning songs. For mammals, new neurons also play an important role in learning. About 1,000 new neurons develop in the hippocampus, a brain structure involved in learning and memory, each day. While most of the new neurons will die, researchers found that an increase in the number of surviving new neurons in the hippocampus correlated with how well rats learned a new task. Interestingly, both exercise and some antidepressant medications also promote neurogenesis in the hippocampus. Stress has the opposite effect. While neurogenesis is quite limited compared to regeneration in other tissues, research in this area may lead to new treatments for disorders such as Alzheimer's, stroke, and epilepsy. How do scientists identify new neurons? A researcher can inject a compound called bromodioxyuridine, BRDU, into the brain of an animal. While all cells will be exposed to BRDU, BRDU will only be incorporated into the DNA of newly generated cells that are in S phase. A technique called immunohistochemistry can be used to attach a fluorescent label to the incorporated BRDU, and a researcher can use fluorescent microscopy to visualize the presence of BRDU, and thus new neurons, in brain tissue. Figure 16.20. This image shows new neurons in a rat hippocampus. New neurons are tagged with BRDU. New neurons tagged with BRDU glow red in this micrograph. While glial cells are often thought of as the supporting cast of the nervous system, the number of glial cells in the brain actually outnumbers the number of neurons by a factor of 10. Neurons would be unable to function without the vital roles that are fulfilled by these glial cells. Glia guide developing neurons to their destinations, buffer ions and chemicals that would otherwise harm neurons, and provide myelin sheaths around axons. When glia do not function properly, the result can be disastrous. Most brain tumors are caused by mutations in glia. How neurons communicate. All functions performed by the nervous system, from a simple motor reflex to more advanced functions like making a memory or a decision, require neurons to communicate with one another. Neurons communicate between the axon of one neuron and the dendrites, and sometimes the cell body of another neuron across the gap between them, known as the synaptic cleft. When an action potential reaches the end of an axon, it stimulates the synaptic membrane of the dendrite or soma of the next cell. The neurotransmitter is released through exocytosis of vesicles containing the neurotransmitter molecules. The neurotransmitter diffuses across the synaptic cleft and binds to receptors in the postsynaptic membrane. These receptor molecules are chemically regulated ion channels and will open, allowing sodium to enter the cell. If sufficient neurotransmitter has been released, an action potential may be initiated in the next cell, but this is not guaranteed. If insufficient neurotransmitter is released, the nerve signal will die at this point. There are a number of different neurotransmitters that are specific to neuron types that have specific functions. The central nervous system. The central nervous system, or CNS, is made up of the brain and spinal cord and is covered with 3 layers of protective coverings called meninges. Meninges is derived from the Greek and means membranes. The outermost layer is the dura mater. The middle layer is the web-like arachnoid mater, and the inner layer is the pia mater, which directly contacts and covers the brain and spinal cord. The space between the arachnoid and pia maters is called the cerebrospinal fluid, or CSF. The brain floats in CSF, which acts as a cushion and shock absorber. Figure 16.21. The cerebral cortex is covered by 3 layers of meninges: the dura, arachnoid, and pia maters. The brain. The brain is the part of the central nervous system that is contained in the cranial cavity of the skull. It includes the cerebral cortex, limbic system, basal ganglia, thalamus, hypothalamus, cerebellum, brainstem, and retinas. The outermost part of the brain is a thick piece of nervous tissue called the cerebral cortex. The cerebral cortex, limbic system, and basal ganglia make up the 2 cerebral hemispheres. A thick fiber bundle called the corpus callosum, corpus meaning body and callosum meaning tough, connects the 2 hemispheres. Although there are some brain functions that are localized more to one hemisphere than the other, the functions of the 2 hemispheres are largely redundant. In fact, sometimes, very rarely, an entire hemisphere is removed to treat severe epilepsy. While patients do suffer some deficits following the surgery, they can have surprisingly few problems, especially when the surgery is performed on children who have very immature nervous systems. In other surgeries to treat severe epilepsy, the corpus callosum is cut instead of removing an entire hemisphere. This causes a condition called split brain, which gives insights into unique functions of the 2 hemispheres. For example, when an object is present to patient's left visual field, they may be unable to verbally name the object and may claim to not have seen an object at all. This is because the visual input from the left visual field crosses and enters the right hemisphere and cannot then signal to the speech center, which generally is found in the left side of the brain. Remarkably, if a split brain patient is asked to pick up a specific object out of a group of objects with the left hand, the patient will be able to do so, but will still be unable to verbally identify it. Each hemisphere contains regions called lobes that are involved in different functions. Each hemisphere of the mammalian cerebral cortex can be broken down into 4 functionally and spatially defined lobes: frontal, parietal, temporal, and occipital. Figure 16.22. The human cerebral cortex includes the frontal, parietal, temporal, and occipital lobes. The frontal lobe is located at the front of the brain, over the eyes. This lobe contains the olfactory bulb, which processes smells. The frontal lobe also contains the motor cortex, which is important for planning and implementing movement. Areas within the motor cortex map to different muscle groups. Neurons in the frontal lobe also control cognitive functions like maintaining attention, speech, and decision making. Studies of humans who have damaged their frontal lobes show that parts of this area are involved in personality, socialization, and assessing risk. The parietal lobe is located at the top of the brain. Neurons in the parietal lobe are involved in speech and also reading. Two of the parietal lobe's main functions are processing somatosensation, touch sensations like pressure, pain, heat, and cold, and processing proprioception, the sense of how parts of the body are oriented in space. The parietal lobe contains a somatosensory map of the body similar to the motor cortex. The occipital lobe is located at the back of the brain. It is primarily involved in vision, seeing, recognizing, and identifying the visual world. The temporal lobe is located at the base of the brain and is primarily involved in processing and interpreting sounds. It also contains the hippocampus, named for the Greek for seahorse, which it resembles in shape, a structure that processes memory formation. The role of the hippocampus in memory was partially determined by studying one famous epileptic patient, HM, who had both sides of his hippocampus removed in an attempt to cure his epilepsy. His seizures went away, but he could no longer form new memories, although he could remember some facts from before his surgery and could learn new motor tasks. Interconnected brain areas called the basal ganglia play important roles in movement, control, and posture. The basal ganglia also regulate motivation. The thalamus acts as a gateway to and from the cortex. It receives sensory and motor inputs from the body and also receives feedback from the cortex. This feedback mechanism can modulate conscious awareness of sensory and motor inputs, depending on the attention and arousal state of the animal. The thalamus helps regulate consciousness, arousal, and sleep states. Below the thalamus is the hypothalamus. The hypothalamus controls the endocrine system by sending signals to the pituitary gland. Among other functions, the hypothalamus is the body's thermostat. It makes sure the body temperature is kept at appropriate levels. Neurons within the hypothalamus also regulate circadian rhythms, sometimes called sleep cycles. The limbic system is a connected set of structures that regulates emotion, as well as behaviors related to fear and motivation. It plays a role in memory formation and includes parts of the thalamus and hypothalamus, as well as the hippocampus. One important structure within the limbic system is a temporal lobe structure called the amygdala. The two amygdala, one on each side, are important both for the sensation of fear and for recognizing fearful faces. The cerebellum, meaning little brain, sits at the base of the brain on top of the brainstem. The cerebellum controls balance and aids in coordinating movement and learning new motor tasks. The cerebellum of birds is large compared to other vertebrates because of the coordination required by flight. The brainstem connects the rest of the brain with the spinal cord and regulates some of the most important and basic functions of the nervous system, including breathing, swallowing, digestion, sleeping, walking, and sensory and motor information integration. Spinal cord. Connecting to the brainstem and extending down the body through the spinal column is the spinal cord. The spinal cord is a thick bundle of nerve tissue that carries information about the body to the brain and from the brain to the body. The spinal cord is contained within the meninges and the bones of the vertebral column, but is able to communicate signals to and from the body through its connections with spinal nerves, part of the peripheral nervous system. A cross-section of the spinal cord looks like a white oval containing a gray butterfly shape. Axons make up the white matter, and neuron and glia cells and interneurons make up the gray matter. Axons and cell bodies in the dorsa spinal cord convey mostly sensory information about from from the body to the brain. Let me try that one more time. Axons and cell bodies in the dorsospinal cord convey mostly sensory information from the body to the brain. Axons and cell bodies in the spinal cord primarily transmit signals controlling movement from the brain to the body. The spinal cord also controls motor reflexes. These reflexes are quick, unconscious movements, like automatically removing a hand from a hot object. Reflexes are so fast because they involve local synaptic connections. For example, the knee reflex that a doctor tests during a routine physical is controlled by a single synapse between a sensory neuron and a motor neuron. While a reflex may only require the involvement of one or two synapses, synapses with interneurons in the spinal column transmit information to the brain to convey what happened. The knee jerked or the hand was hot. Figure 16.23, a cross-section of the spinal cord, shows gray matter, containing cell bodies and interneurons, and white matter, containing myelinated axons. The peripheral nervous system. The peripheral nervous system, or PNS, is the connection between the central nervous system and the rest of the body. The PNS can be broken down into the autonomic nervous system, which controls bodily functions without conscious control, and the sensory somatic nervous system, which transmits sensory information from the skin, muscles, and sensory organs to the CNS and sends motor commands from the CNS to the muscles. Figure 16.24. In the autonomic nervous system, a preganglionic neuron originating in the CNS synapses to a neuron in a ganglion that, in turn, synapses on a target organ. Activation of the sympathetic nervous system causes release of norepinephrine on the target organ. Activation of the parasympathetic nervous system causes release of acetylcholine on the target organ. The autonomic nervous system serves as the relay between the CNS and the internal organs. It controls the lungs, the heart, smooth muscle, and exocrine and endocrine glands. The autonomic nervous system controls these organs largely without conscious control. It can continuously monitor the conditions of these different systems and implement changes as needed. Signaling to the target tissue usually involves two synapses: a preganglionic neuron originating in the CNS synapses to a neuron in a ganglion that, in turn, synapses on the target organ. There are two divisions of the autonomic nervous system that often have opposing effects: the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is responsible for the immediate responses an animal makes when it encounters a dangerous situation. One way to remember this is to think of the fight or flight response a person feels when encountered by a snake. Snake and sympathetic both begin with S. Examples of functions controlled by the sympathetic nervous system include an accelerated heart rate and inhibited digestion. These functions help prepare an organism's body for the physical strain required to escape a potentially dangerous situation or to fend off a predator. Figure 16.25. The sympathetic and parasympathetic nervous systems often have opposing effects on target organs. While the sympathetic nervous system is activated in stressful situations, the parasympathetic nervous system allows an animal to rest and digest. One way to remember this is to think that during a restful situation, like a picnic, the parasympathetic nervous system is in control. Picnic and parasympathetic both start with P. Parasympathetic preganglionic neurons have cell bodies located in the brainstem and in the sacral, toward the bottom, spinal cord. The parasympathetic nervous system resets organ function after the sympathetic nervous system is activated, including slowing of heart rate, lowered blood pressure, and stimulation of digestion. The sensory somatic nervous system is made up of cranial and spinal nerves and contains both sensory and motor neurons. Sensory neurons transmit sensory information from the skin, skeletal muscle, and sensory organs to the CNS. Motor neurons transmit messages about desired movement from the CNS to the muscles to make them contract. Without its sensory somatic nervous system, an animal would be unable to process any information about its environment, what it sees, feels, hears, and so on, and could not control motor movements. Unlike the autonomic nervous system, which usually has two synapses between the CNS and the target organ, sensory and motor neurons usually only have one synapse. One ending of the neuron is at the organ, and the other directly contacts a CNS neuron.